Hello, my name is Marcio Moraes and I'm a full professor at the Universidade Federal de Minas Gerais. Núcleo de Neurociências is where I work, and you can find more about us if in this link right here, if you're interested. I'm very thankful for the opportunity to speak at Falan, alongside distinguished colleagues that work within the field of neuroscience in Latin America. Well, let's get to the point. Our research interests encompass the structural and functional architecture of the brain. More specifically, how the brain encodes information within the neural networks about the world that surrounds us, and how it processes that information to output appropriate behavior. Well, I'm pretty sure I have narrowed down to the whole field of neuroscience by now. But anyhow, one way to frame this problem is to think about the 80 to 100 billion neurons in the brains in terms of graph theory. Leo and colleagues have provided us with a nice review about this subject. So the whole idea here is, suppose that I have all neurons in the brain, like 1, 2, 3, 4, up to 80 billion, represented by these nodes. So let's call these a node. And the connections between the nodes, we're going to call edges, actually shows how these neurons are interconnected. So we would actually be able to represent this by a matrix that has 80 billion columns, billion, and 80 billion rows. And each one of these elements in this matrix represent the connections between the neurons. Well, in fact, by using diffusion imaging and structural or MRI, we have shown that the organization is most likely in the brain to have strong interconnectivity between groups of neurons. And these groups of neurons send projection to other groups of neurons to transfer local processing to processing pockets within distant parts of the brain. So what I'm trying to say, translating this, is if you look at this matrix right here, if every single neuron would connect to every other single neuron, I would expect this matrix to be completely full like this. But it's not. I mean, if you look at it, it actually has like pockets of information, of connections, showing that perhaps it's not that every single neuron connects to every other neuron. Well, if you think about this, it makes perfect sense. I mean, it's not so far-fetched if you imagine that the huge spatial toll that this would take, and especially metabolic toll, if you have to connect every single neuron to each other. It would be physically impossible. You would not have space to put all this white matter. So other architectures are most likely in the brain. One such arrangement is called small world networks, and it fits perfectly with the idea of modular pockets of information, transfer information to other pockets that I have just discussed. Highly interconnected pockets with distant connections among these modules through communication hubs. So, in fact, if you look at these small world topology, they are usually accompanied by the presence of modular structure and hub nodes, which seems to optimize both the information segregation within a specific module. But not only that, but it seems to be ideal in order to integrate information with other networks, meaning that not only it has good clustering coefficients, it clusters information in modules like this, but it also maintains the idea of short distances between these modules to transfer information among the modules. Now, if this is the case, then we are not, the brain architecture will be somewhere around here in the increasing randomness of its interconnections. Now, forgive the pun here, but if small world networks are the case, how do we transfer spatial temporal discharge patterns from one module to the next? Well, what I mean to say is, suppose that this neuron fires in this one and then this one, so in this order, and this actually is a pattern that represents something that's happening in the outside world. How do you transfer this information from module A to B? This is a big problem, and I would definitely say that we might be able to solve this problem to think about oscillations in the target module that will make certain neurons more prone to discharge at one specific time, meaning that Suppose that in a particular point of a slow wave that's passing through these neurons are with low probability of discharge, while these neurons are with high probability of discharge. So the encoding happens not where, but when this hub discharges. If it discharges at this specific point of the wave, it would probably recruit this guy. But if it discharges, I don't know, at another point of the wave, it could recruit another neuron that's more prone to discharge. So within a period of oscillation, that would be where the information of a specific neuron in module A is supposed to recruit specific neurons in module B, which means that this would be some sort of temporal multiplexing. Well, the concept of oscillations is not new in neuroscience. And actually, what I'm talking about here is definitely has been proposed elsewhere. If you look at Busacchi, if you look at Varela, they actually provide, and there's a lot of evidence showing that phase encoding of specific discharge of gamma oscillations around slower oscillations like theta oscillations, they actually encode information about what the animal is about to do or specific behavioral tasks. Anyhow, how do we test this? So how do we go about and actually design an experiment that would actually be able to test this? So how do we show how temporal coding influences this dynamical connectivity between modules? Well, one idea that we had is to get a specific theta oscillation as a time frame. So this would be 100 milliseconds. So within that time frame of 100 milliseconds, we had two different patterns, both pattern A, which is this pattern, and pattern B, which is this pattern. And we tried to design a protocol that would actually get the animal to learn that pattern A, he's supposed to do something, but not with pattern B. And by applying pattern B, he would do something and not with pattern A. But in order for this not to have any idea that we are recruiting different modules or modules in different intensities, we applied the stimulation to what we thought was a good node, which is the amyloid complex. And we used this, we applied to this node the exact same amount of stimuli, so 6 stimuli in both patterns. The only thing that changed, so we applied to the same place with the same amount of stimuli, the only thing that changed is the temporal organization of these patterns. And we changed the temporal organization. So the whole idea here is we had two groups of animals, mainly. So this is the work done by Flávio Afonso Gonçalves Mourão. He's a postdoc resident at Núcleo de Neurociências. And we published this in Cerebral Cortex in 2015. But what Mourão showed is that if we had two groups, so let's call group A the group that has pattern A paired with the foot shock, and group B that has pattern B paired with the foot shock. Now, it's important to highlight that both groups received both patterns A and B. The only difference between them is that the group A had a foot shock paired with pattern B and not paired with pattern A, but not paired with pattern B. And group B had a foot shock paired with pattern B, but not paired with pattern A. But they received pattern A and B both. And the whole idea here, so this is a control only, but the whole idea here is when we tested and we applied pattern B to the amyloid complex during the retention test phase, what happened, and so this is a normal, common fear conditioning test. What we saw is that only group B actually freezed, meaning that it recognized. But when we applied pattern B to group A, since it was not paired with pattern B, it did not freeze. It did not freeze. But 24 hours after that, just to make sure, we applied now pattern A. And group B that responded so well with pattern B, when we applied pattern A, group B did not respond or respond close to what controls would, unpaired groups would. But pattern A now, pattern A, well, this one had a lot of freezing. Just to make sure again, 24 hours after that, so 72 hours after the first test, we applied pattern B again. And when we applied pattern B, again, as happened in the first day, group B had a lot of freezing, and group A didn't have much freezing. Now, the whole idea here, and we tested this with other metrics too. I'm not going to get into it because of the lack of time, but the whole idea here is what neurons or what modules were actually active during that time. And if you looked at the last stimuli, we had B pattern applied. So this is everything that happened after we applied pattern B. By applying pattern B, you can see that only the B group, the one that was paired to it, actually had big recruitment of the dorsal medial hypothalamus and the ventral medial hypothalamus. Well, these are output structures to the fear behavior. So these are output pathways to the to sympatho-excitatory fear behavior. So this makes sense. But the interesting thing is the group A. So remember that group A received at this time the B pattern stimulation. It had no recruitment of the hypothalamic structures. Now, if you look at the amygdaloid complex, we saw kind of the same thing with group B, but here's the interesting data. If you look at the prefrontal cortex, no, it was only really recruited. So the prefrontal neurons or the prefrontal module was only recruited when the animal did not know that he had to have a freezing behavior. So he was like not sure what he had to do in terms of output of behavior. So in this case, you had a huge recruitment of the prefrontal cortex, which is completely consistent with what we think is the role of prefrontal cortex inhibiting the conditioned response of amygdalo hypothalamic discharge. We tried to put this idea of temporal patterns to good use. And the whole idea to put this to good use was to use this in epilepsy. So suppose that you could think about epilepsy as a pattern of stimulation of ictal discharges. What would it take to disrupt that pattern, to disrupt those connections that recruit more and more by hypercoupling different modules? Well, one of the ideas is perhaps we could mess up these hyperconnectivity by using different stimuli. So this is the pattern that is a discharge pattern, the normal discharge pattern that you would see in epileptic animals with PTZ-induced seizures. And this is a completely, it's a kind of a messed up temporal code. So the whole idea here is this, in what we are thinking, would actually promote or facilitate the coupling between networks, while this would disrupt it. And this is exactly what we found. We saw that the periodic stimulation that would actually resemble the firing pattern, temporal pattern of discharge of the ictus, actually was pro-convulsant, while the messed up temporal code that we used during ictogenesis actually was anti-convulsant. This is the work done by Vinícius Rosa-Cota, which is a professor now, so Professor Vinícius Rosa-Cota at the Universidade Federal de São João. He has his own research group called LINS. We actually tested this. This is Michel Bernanos. We tested this with MRI studies. And exactly the same with the periodic stimulation. Here's the amygdaloid complex highlighted. With the non-periodic, and this was quite interesting, we saw a huge recruitment of the prefrontal cortex also. But as we saw with the behavioral data, periodic stimulation increased activity in the ictogenic during the ipsilateral side of stimulation during PTZ seizures, and non-periodic blocked where the recruitment of those modules during ictogenic. So we saw that we could interfere with the coupling, but can we predict if something like that's going to happen? So I'm going to call this active probing. So in order to do that, and I'm going to go really fast by this, we had both mathematical models coupling population A and B, and we had a biological model using PTZ to couple to use for predicting. And both DBS and this mathematical model of DBS we were actually able to show that an abnormal coupling between modules was happening. And I'm not going to have time to go into this in detail, but we actually did this both in animal research and clinical data using a sensory stimuli, that's auditory steady state stimulation. And we showed that we could actually predict, so we have pretty good indication that we can predict surgery prognosis, and we actually saw a very good chance of predicting this hypercoupling in epileptic animals. Well, I'd like to thank my collaborators for this, and please feel free to contact us in using this link. Thank you very much.